Hi, Miss Salties. Hope you're doing well in this difficult time. So today, Sanya and I will be talking about telomeres and the aging process. So there's going to be four aspects that we'll be introducing in this topic. First, I'll be talking about what telomeres are and their functions. Second, the factors that impact telomeres length and way to slow down telomeres. Sanya will be also introducing two new points about techniques and cost, and last but not least, the ethics and social issues. Okay, so the first is what are telomeres and their functions? So getting older is a part of life, and understanding why we age or what causes aging is a puzzle that scientists are still trying to solve. So our theory suggests that the aging process is at least particularly linked to our telomeres. So there's always these little protective caps at the ends of our DNA molecule that make up chromosomes. So their job is to stop the ends of chromosomes from fraying or sticking to each other, much like plastic tips on ends of the shoes. Yeah, we can move on. So cells use a special enzyme called telomerase, which helps the cell to divide and lengthen the telomeres, and very low concentration in somatic cells. So somatic cells are just like any cell of a living organism other than cells. So aging cells, aging bodies. Okay, so possible results of the problem. So loss in genetic information. Why is it bad? Because the telomeres are usually at the end of your DNA molecule. So what it does is after your cells are replicating many times, your chromosomes actually shorten. So what is actually shortening is your telomeres. But as you age, you don't have as much telomeres left. So then you're actually cutting down genetic material, so you're losing DNA because your chromosomes are getting shorter and there's no more telomeres to protect that, so it's just cutting down DNA. Second is defective proteins and dysfunctional enzymes, death of cells, disease and death. So factors that impact telomeres length and need to slow down the shrinking. Okay, so I'm just going to talk about the primary impact effects on it. So stress is one because it releases hormones by the adrenaline gland, and the level of antioxidant proteins are reduced and can induce DNA damage and therefore expedite telomere shortening. Smoking is also associated with accelerated telomere shortening. So the dosage of cigarette smoking is shown to correlate. Telomeres also in obese women have been shown to be significantly shorter than those in women. So that's just not just in women, just in people generally, but it's mainly caused women. And a lack of exercise, because exercise is very important because it reduces inflammation and oxidative stress. So you can see this correlation where it all links back to oxidative stress. Okay, so ways to slow down that. So diet. Diet is very important. So similarly, the woman who consumed a lacking of antioxidants had shorter telomeres, and that also had a moderate risk for, a high risk for development of breast cancer. So yeah, and these are just things that are listed of what you should eat and what you should not eat to slow down the process. Second is exercise. So exercise is very important. It can reduce harmful fats, help mobilize waste products like vascular inflammation, and it leads to reduced stress. So again, stress. That's a very like big factor when it comes to certain telomeres, and is the preservation of DNA and telomeres. So it just there is a study that shows that people who participate in high levels of activity have significantly longer telomeres than those who did low or activity. Stress management. When you're stressed, your body releases hormones that can also cause oxidative stress. So that can also result in more shortening. So again, eating healthy, staying active and fit, these are always very good for our body and also for our telomeres. And again, less exposure to pollution and toxic environments because Investigators actually have found out that telomeres the traffic police oxidate shorter within each age relative to telomeres length like normal oxygen.
Okay, so Rhea mentioned ways that telomere length is lengthened, and she talked about things that we can do to shorten this process. So today I'm going to be talking about the technique and economic implications, and I'm going to be expanding on that and talking about what scientists are currently doing. So as of right now, there's a test called the FlowFish test, and that stands for flow cytometry and fluorescence in situ hybridization. And so what that does is specifically measures telomere length in each cell of a patient's blood sample. So this is a Canadian innovation, and the test was developed in about the year 2006 by Peter Lancer. And in the test, scientists poke tiny holes in the nucleus of each cell, and what they do is they slip in these fluorescent labeled DNA that attach to the telomeres, and they then use this method called flow cytometry, which is in the name, and it's just a method in simpler terms which can count specific cells and identify specific components within the cells. So they use this method to count the fluorescent signals in the cell one at a time. So what does that leave us with? So it basically gives us this test gives us the patient's median telomere length, and it's then virtually displayed on graphs against a percentile. So if somebody's below the first percentile, that's a case where you'd be concerned, because that means 99% of that age range has a different telomere length. Now, in terms of cost, so for this specific test, it can cost about $400, and cost can vary depending on how it's retrieved. So, if you're obtaining this test through a physician, it might be more, because you're also paying for the time that they're spending on that. It also might be less if you're investing in a commercial home kit. So if you buy it from a business, it might be obviously lower, because those aren't FDA approved yet. So you might be sacrificing the accuracy of the results. So if you want to buy a cheaper kit for telomere length testing, you'd probably also be getting a lower accuracy. So now for ethics and social issues, I'm going to briefly go over this. So something that we have to consider is that we're still unsure of how reverse aging will affect the brain of a human that we're going to be giving these services to. So patients might still develop the same psychological problems that another elderly person that isn't receiving this treatment is. So that can be like isolation or depression. To consider as well is that by providing something that will lengthen somebody else's life, it might go against somebody's core beliefs and religion, because by doing this, we're putting the hands of somebody's life in doctors and scientists, and it's basically going against nature, because you're lengthening somebody's life. So yeah, that might be concerning. But something to look on the bright side is that we could develop a pill and make it more accessible to a wider range of people. We'd also be contributing to academia. It's a new invention, it's very exciting. And also another thing is potential biological immortality. So you'd obviously be living longer, and that for many is a plus. And they'd be comfortable in living their life, because they know they're going to be around for a longer period of time. Here's a statistic. So in 2014, over 6 million Canadians were aged 65 or older, representing 15.6% of Canada's population. And by 2030, in less than 2 decades, seniors will number over 9.5 million and make up 23% of Canadians. So what does this mean for society? So let's say this does work out. seniors will be living longer, and even people in like the middle age, so like maybe 40 to 45, they might be investing in this, which will probably mean that they will have their jobs for longer. So that means there's going to be less opportunities for the younger working class. And there also might be a decline in some businesses and an increase in businesses that are catered to adults and seniors. So, for example, in urban planning, we'd see a lot more hospitals. We'd also see a lot more clinics that are providing this test. And we'd probably see less daycares. So here's Ria's works cited. Here's my works cited. So that is all for our presentation today. Thank you so much.
Bye.